Hello friends, it's me Nishi here with a new video on molecular basis of inheritance tricks. So, if you haven't subscribed to my channel, please do that now and press the bell icon to get notified whenever I post new videos. And you can have a look at the description box to know which all topics are included in this video. So without any further delay, let's get started. First topic is nitrogenous base. Many of you may know the trick of this, but still I am telling you that purine. First is purine. It consists of adenine and guanine, and pyrimidine is of cytosine and thymine, and in case of it is uracil. In case of RNA it is uracil. So the trick is go add purifier and cut pyrimid. So go add purifier means go for guanine, add adenine, purify, that is purine. So purine consists of guanine and adenine. In case of pyrimidine, it's cut pyrimid. C for cytosine, U for uracil, and T for thymine. Then pyrimid for pyrimidine. So you can learn it as cut pyrimid in gap. Gap for guanine, adenine, and purine. Okay, as you wish. You can learn either of this. And the next thing is A, that is adenine is with thymine with a double bond, with double H bond. Okay, with double hydrogen bond. Okay, you can learn it as at. So at A bonds with T, that is with double hydrogen bond. Okay, so next topic is, and before that guanine is bonded with cytosine with triple. Okay, when A is double with hydrogen bond with T, then G will be, of course, G will be of triple bond with C. Okay, you can learn any of this. Now the next topic is structure of polynucleotide chain. Okay, so structure of polynucleotide chain. We will be learning a story for this. Okay, it's a weird story, but still you can understand. You can just imagine as a movie. Okay, so someone was pushed into police station with notebook. So imagine this someone. Okay, he's pushed into the police station. Okay, imagine this is a police station with notebooks. Okay, he was pushed into a police station with a notebook. So someone, that is nucleoside, S for someone, nucleoside, pushed into police station, pentose sugar, police station with notebook, that is with means plus, NB, that is nitrogenous base. So when it is nucleoside, it is of pentose sugar plus nitrogenous base. And notebook gain learning. So what happened? Because of that someone was pushed into the jail with, that is police station with notebook, it helped him to gain knowledge, that is gain learning. So he learned something, that is notebook gain learning, that is N-glycosidic linkage. So the linkage between these two, police station and notebook, is N-glycosidic linkage. And after few months, that someone was badged a thief. So after a few months, what happened? That someone was badged as a thief. So he was a thief. So thief is someone with PG, that is postgraduate. Okay, so thief is someone with postgraduate certificate, that is PG, that is nucleotide, tide for thief. Okay, thief equal to nucleoside, that is someone, plus phosphate group, that is postgraduate, PG. Okay, so the linkage is passed exam. So what happened? Because postgraduate, how did he become the postgraduate? What is the link between someone and PG? It is because he passed an exam. So passed exam, that is phosphoester. Many will be confused that whether the linkage is phosphoester or phosphodiester. So this trick gonna help you a lot. That is passed exam. Someone became a postgraduate by passing exam. Pass exam. So linkage between nucleoside and phosphate group is phosphoester. Next is, this thief was made to meet another thief as both passed double exam. So both of them passed double exam. So both of them met each other. That is, the linkage between two nucleosides is through phosphodiester linkage. That is passed double exam. So phosphodiester linkage. This was our trick, and I hope this helped you. Next is backbone of police committee. Okay, the backbone of police committee is, ignore the spelling mistake. Okay, I was in a hurry when I wrote this. Okay, so backbone is police committee, that is polynucleotide chain is SP, that is sugar and phosphate. So backbone of police committee is SP, that is sugar and phosphate. Next topic is Chargaffs Rule. Okay, Chargaffs rule is that amount of adenine equal to thymine and guanine equals cytosine. So the trick to learn this is aryl equal to Tide. You all know this, right? A washing powder, aryl and Tide. So both of them are similar. That is washing powder. So aryl equal to Tide. That is A equal to T. And what happens when A plus G by T plus C equal to 1. Okay, if you know the basic mathematics, then you can easily write this. That is A plus G by T plus C equal to 1. That's Chargaffs rule. Next is packing in eukaryotes. Okay, how is the DNA packed in eukaryotes? So histone octamer plus DNA. Then it is first is histone octamer plus DNA, the small unit, and then little big, that is nucleosome. Then all the nucleosomes constitute the chromatin, and all these chromatin constitute the chromatin fiber, and which is of chromosome, which is again into chromosome. Okay, so histone octamer, the histone octamer looks like this, okay, like a spear, and plus DNA. Okay, this is H1, and a DNA is packed like this. Okay, and this is what this is one histone octamer plus DNA. When one or two histone octamer, that is nucleosome, and all these nucleosomes constitute the chromatin. Like that it goes on. And the trick for that is HT is nice. HT equal to nice. HT, that is histone octamer plus DNA. HT is nice. Nice for nucleosome, and chromatin or chromatin fiber and chromosome. The easy trick is in alphabetical order. First is chromatin, because in chromatin after M there is A, which comes first. Then here chromatin fiber. Okay, chromatin same A, but then there is this fiber. Okay, so it comes second. Next is chromosome, O. After M it is O, which comes last. Okay, so first chromatin, then chromatin fiber, then chromosome. Okay, that's the trick of packaging in eukaryotes. In typical nucleus, some region of chromatin are loosely packed. So those regions which are loosely packed, that stains light, and it's called euchromatin, and which are densely packed stains dark and are called heterochromatin. So the easy trick is densely packed means dark, loosely packed means light, LLDD. And you have to know which is euchromatin and which is heterochromatin, right? For that we will look this trick, that is HT, that is heterochromatin, dark. So the other will be, of course, light will be euchromatin. So heterochromatin is dark, stains dark, and is densely packed. Okay, the next topic is transforming principle. So in transforming principle, the trick is thief pleaded SP and freedom given. So we looked about this thief, right, in the previous trick. So that thief pleaded SP. So he pleaded the SP and freedom was given to him. So thief pleaded SP and freedom given. Okay, and this is not that same SP, that is sugar and phosphate, but this is someone else. Okay, let's look who it is. So thief pleaded, that is transforming principle, to know which trick that belongs to. So transforming principle, and SP, that is Streptococcus pneumonia, and freedom given, that is Frederick Griffith. So Frederick Griffith did this transforming principle experiment with Streptococcus pneumonia. So transforming principle was did by Frederick Griffith with Streptococcus pneumonia. Thief pleaded SP and freedom given. So now S strain bacteria has this polysaccharide coat which makes it smooth, and R strain bacteria have this rough coat. Okay, it's rough, but S strain is smooth. You know this, S for smooth and R for rough. Okay, when this S strain is injected into mice, the mice die. So easy trick to remember this is when you walk in a smooth surface. Okay, if someone walk in a very smooth surface, it's slippery and they will fall down. And what happens when they fall down? That may lead to death, even death. Okay, so what happens in smooth surface? Mice dies. That is, in smooth strain bacteria is injected into mice, the mice dies. Next is rough strain injected into mice live, because due to friction we can walk in rough places, right? This is not the real reason, but I am just making it more simple for you with this trick. So rough strain into mice live. Next is S strain is heat kill. When the S strain is heat kill injected into mice live. So when the S strain is heat killed and is injected into mice live. Okay, the real reason is when the S strain is injected into mice, the mice live, and we are not learning this short trick for this. And next is S strain plus R strain, that is S strain which is heat kill. This one plus R strain which is live. It is injected into mice, the mice die. So what happens? Something from the R strain goes to the S strain, which makes it virulent, that is it makes the mice die. So we don't know what it is, but they know there is something present in the R strain which is present in the S strain. Even after heat kill it's present, but this R strain gives to the S strain. Okay, the next topic is DNA as genetic material. Okay, DNA is genetic material. There will be two tricks for this, that is busy thief presently in Oxford Medical College. So now that thief, the same thief, okay, we are learning about this thief for so long. Actually now that thief is busy thief presently in Oxford Medical College. Busy for biochemical characterization. So biochemical characterization of transforming principle. Thief presently, that is transforming principle. So busy thief presently. So biochemical characterization of transforming principle was done by Oswald Avery, that is Oxford, and medical for MacLeod McCarthy, and college for Colin MacLeod. So OMC, that is Oxford Medical College. Okay, busy thief presently college. The next trick is thief 2 busy entertaining all her friends in marathon race or chase. You can learn it as race or chase. That is there were two thieves, right? The both thieves met each other and they passed double exams. That thief. Okay, the second thief, that is thief 2. So thief 2 for T2, busy for bacteriophage, entertaining, that is for experiment. Okay, so in the T2 bacteriophage experiment was done by all her friends in marathon race or chase, that is Alfred Hershey and Martha Chase. Okay, Alfred Hershey and Martha Chase. So thief 2 busy entertaining all her friends in marathon race or chase. You can learn it as race or chase. Now the next topic is semi-conservative DNA replication. The trick for this is Supreme Court welcome Friday Max exam when Taylor and colleagues visited France. Okay, so the Supreme Court welcomed the Friday Max exam. When did this happen? When Taylor and his colleagues visited France, and his colleagues visited France. Okay, so Supreme Court, that is semi-conservative of Watson and Crick. So semi-conservative of DNA replication was introduced by Watson and Crick. Okay, welcome means we are introducing something, right? By that also you can learn it. Watson and Crick. Supreme Court welcome Friday Max exam. That is Franklin Stahl and Matthew Meselson did the experiment. Okay, the semi-conservative DNA replication experiment was done by Franklin Stahl and Matthew Meselson. So Supreme Court welcome Friday Max exam when Taylor and his colleagues visited France. So Taylor and his colleagues did the same experiment with Vicia faba with France. Now our next topic is enzymes required for DNA replication. The trick for this is SPB in top of hell practicing for Linga dance. So this is there's this man called SPB. He is in the top of hell, or you can learn it as hell as you wish. Okay, he's in the top of hell practicing for Linga dance. So he is practicing for a dance from movie Linga. So SPB in top of hell practicing for Linga dance, or you can learn it as hell, your wish. Okay, SPB for SSBPs and top for topoisomerase, hell for helicase, practicing for primase, and for phosphorylase, and Linga ligase, dance for DNA dependent DNA polymerase. Okay, so these are the enzymes required for DNA replication. Our next topic is transcription unit. So there are three regions in transcription unit, that is promoter, structural gene, and terminator. Easy trick is pest. Okay, and PST. After P there will be QR and ST. So E is silent here, that is we don't need E here, that's why. Okay, pest. After P, ST, PST. Promoter, structural gene, and terminator. Now the next topic is Transcription in prokaryotes. So here we are going to learn a very interesting story. The characters included are RNA, that is RNA polymerase. Then we have SIMA, that is sigma factor, ROHIT, that is Rho factor, and RANA for RNA. These four are the characters. Okay, we are going to see a movie or a story. Now the equipments, that is in this movie or in this story, there are certain equipments, right? That is DNA template or transcription unit, that is called the train here. So train is DNA template or transcription unit. So you can imagine that this story is gonna take place in a nucleoside triphosphate is entry ticket, because entry and ticket, that is triphosphate ticket. Okay, entry ticket is nucleoside triphosphate. You can remember like that. So the story begins. Okay, this is a train. Just think this DNA helix as a train. Okay, and what happens? There is this promoter region, and this is the train. Okay, I wrote it as bus, but it's actually a train. Okay, this train. So what happens? Rena enters the train using entry ticket. Okay, this girl Rena, she is Rena. Okay, she enters the train using entry ticket, that is nucleoside triphosphate, with the help of sigma, that is sigma factor. This is SIMA. So Rena is entering the train with entry ticket with the help of SIMA. Using entry ticket, Rena enters the train with the help of SIMA. So RNA polymerase is going into this DNA helix with the help of sigma factor, using nucleoside triphosphate as substrate. Okay, so Rena has entered the train using entry ticket with the help of SIMA. Then after the train, that was the initiation step. So elongation step is after train starts, SIMA leaves the train and Rena travels independently. So when this train starts, what happens? SIMA, that is sigma factor, leaves the DNA helix, that is a train. So after train starts, SIMA leaves the train and Rena travels independently. So now the RNA polymerase is independent of anyone. So it is traveling independently. Next is termination. In this termination step, what happens is, in the Rena meets the small kid Rana. So RNA polymerase, that is Rena means the small kid Rana RNA with the help of Rohit. So this guy called Rohit, he is waiting for her in the railway station to pick up Rena from the train. So with the help of Rohit, she leaves the train and brings Rana along with her. She lives with Rohit. Rana is independent now. So what happens when after coming to this point? What happens? Rena is coming out of the train with the help of Rohit. So Rohit and Rena are together now, and she leaves this kid Rana alone. So now RNA, that is Rana, is independent. Now if you see this in the textbook, it will be very easy for you if you remember this story. Our next topic is transcription in eukaryotes. Okay, so RNA polymerase 1 transcribes rRNAs, that is 28S, 18S and 5.8S. Okay, you have to remember this, 28S, 18S and 5.8S. Then RNA polymerase 2 transcribes a precursor of mRNA called heterogeneous nuclear RNA or hnRNA. And RNA polymerase 3 transcribes tRNA, that is 5S rRNA and snRNAs. Okay, small nuclear RNA. So 5S RNA, rRNA, srRNA. Okay, and snRNA, srRNA and snRNA. So the trick is rhinotarsen. So rhino is with tarsen. So rhinotarsen, and that is rhino, which comes first, that is RNA polymerase 1 for rRNA, and hnRNA, that is second. Okay, second one that are transcribed for hnRNA. And now the third one. Okay, third one transcribed for tRNA, that is tarsen T. Then rRNA, that is 5S rRNA, 5S rRNA. Then ZAN, that is, we can be say this as S also, right? So snRNAs. The next is post-transcriptional modification. Okay, in eukaryotes, what happens is the primary transcripts are non-functional with both the coding regions, exons, and non-coding regions, introns present in RNA, and this is called hnRNA. So easy trick to remember which is that coding and which is non-coding is. In non-coding there is 2N, right? N-O-N. So non 2N. And intron also have 2N. N-I-N-T-R-O-N. So 2N means press on in non-coding and intron. So intron is non-coding. And exon is coding region. Now the next one is capping, tailing and splicing. 3 of it happens in this HNRNA to make it an mRNA. So capping. It is unusual nucleotide methyl guanine triphosphate is added to the 5' end of HNRNA. So you can remember this as CM. Okay, CM, that is capping methyl guanine triphosphate, CM. Next is tailing. That is adenylate residues about 200 to 300 are added at the 3' end in a template independent manner. So you can learn it as AT. For 3, that is 3'. So tailing for 3, that is 3'. So T for 3, that is 3' end. So tailing is done in the 3' end while capping is done in the 5' end. So tailing, T for 3. Next is splicing, that is introns are removed and exons are joined to form mRNA. In splicing, introns are removed and exons are joined. So we want to make it coding, right? So that's why this coding regions, that is exons, are joined and this non-coding introns are removed. Next, know that structural gene in prokaryotes is polycistronic and continuous, while structural gene in eukaryotes is monocistronic. Trick to remember this is prokaryotes P, polycistronic P. Okay, and in eukaryotes it's monocistronic. You can learn it as me, that is monocistronic eukaryotes, ME. Next topic is stop or terminating codons. Okay, stop or terminating codons. So the 3 are UAA, UGA and UAG. Okay, so first what you have to do is, first you have to write U. In 3 of the cases, the first thing is U. U. Okay, now we have to write A 4 times and G will be of 2 times. So first thing is we write A 2 times here and there will be A 4 times, right? 1, 2, 3 and here like this, AA. And here G. Here also G. Is very simple. So UAA, UAG, UGA. So A will be coming 4, then G 2 times and U 3 times. Okay, if you remember like this, it will be very easy for you to learn. Next is lac operon. So lac operon, we are going to learn another story. This will be a story which happens in every household. So first we will be looking at the switched off condition. Imagine this as your room. Okay, this is your room and this is your light switch. Okay, light switch, and this is your mom. Your mom is the repressor here. So what happens? Your mom is the repressor, and what happens here is this repressor binds to this switch. That is, your mom comes to the switch and she offs the light. So what happens? You want to ask permission to your big sis, that is a big girl. Okay, she's a big girl in your house according to you. So you are asking your big sis for tripping, but as your mom switches off the light, you can't do that. So your mom comes and switches off the light. So you are unable to ask your sis the permission that whether you can go for tripping or not, and whether she permits or not. You are not able to know that since your mom switched off the light. That is, there is an absence of inducer. That is, no one is coming there to make your mom stay there itself without making her to off the switch. The next one is switched on condition. In this, what happens is when your mom comes to off the switch, there will be a relative aunt. She comes and talks to your mom. And what happened? Your mom is not offing the switch now because she is busy talking with your relative aunt. And now you ask your sister if you can go for a tripping, and big girl permits tripping. That is big girl, that is beta galactoside, permits, that is permease, tripping, that is transacetylase. So big girl permits tripping. So these were the tricks for this chapter. If you like my video, then please do subscribe to my channel and press the bell icon to get notified whenever I post new videos. Do share this video with your friends. Thank you and have a nice day ahead.